Bismillah ar-Rahman ar-Rahim. Today's presentation is basal metabolic rate. What is basal metabolism? Minimum energy cost of living rather than the heat required to maintain body temperature. So, very simply, it is the minimum energy cost of living. The turnover of energy in a fasting and resting organism, usually using energy solely to maintain vital cellular activity, respiration and circulation as measured by the basal metabolic rate. Basal metabolic rate is the number of calories per unit time required to keep your body functioning at rest. BMR is also known as your body's metabolism. Basal metabolic rate introduction with the human as exemplary organism. The basal metabolic rate BMR is the amount of energy that is expended at rest in a neutral environment after the digestive system has been inactive for about 12 hours. It is the rate of one's metabolism when waking in the morning after fasting during the sleep. The BMR is enough energy for the brain and central nervous system, heart, kidneys, liver, lungs, muscles, sex organs and skin to function properly. People who are overweight or obese do not necessarily have a slow BMR. In fact, their BMR is usually faster to accommodate for extra fat and for their body to work harder to perform normal body functions. Building lean muscle mass can increase BMR, but there is a limit for both men and women as to how much lean muscle mass can be built. Some supplements may increase BMR, but also only to a limit, and they may have a serious side effects. Expending extra calories through increased physical activity is the most sensible way to increase metabolism. When a person diets, BMR slows down to conserve energy and protect vital organs. A regime of reasonable dieting with increased exercise maintains or increases BMR and promotes weight loss and weight maintenance. It all depends on calories and caloric balance. Basal metabolic rate in animals. Now, with the perspective of animals. In the last slide, we discussed the perspective of human being as an exemplary organism. Now, starting with the basal metabolic rate in animals. First of all, we shall discuss about the historical perspective. In 1766, Carl Shelley discovered oxygen gas and named it fire air. 2 years later, Joseph Priestley independently isolated the same gas, calling it dephlogisticated air. Antony Lavoisier, regarded as the founder of modern chemistry. Lavoisier should be regarded as the father of the basal metabolic rate as well, because in his work with his wife as a major collaborator, Lavoisier showed that animal respiration is the combination of oxygen from air with carbon and hydrogen from the animal's body, which he recognized came from the food to produce water, fixed air, carbon dioxide and heat. He showed that the rate of oxygen consumption was influenced by the number 1, consumption of food, number 2, environmental temperature, number 3, the performance of muscular work. Lavoisier measured the minimal metabolic rate in the resting post-absorptive state, which was probably the first measurement of BMR after 1989. In 1895, A. Magnus Levy showed that secretions from the thyroid gland stimulated the metabolic rate of humans. Until the development of radioimmunoassays in 1960s, for the measurement of blood thyroid hormone concentrations, the measurement of BMR was an important clinical measurement used to assess thyroid status of individual humans. This clinical context was a major influence on the development of strict conditions for the measurement of metabolic rate and the refinement of the BMR concept during the 20th century. During this period, BMR was also increasingly used to compare metabolic activity among various mammals and birds. However, The detailed conditions prescribed for the correct measurement of BMR of humans were not easily translated to the measurement of BMR in animals. The concept of standard metabolic rate (SMR) was also developed to analyze the bioenergetics of ectotherms. It is related to BMR and includes the specification of temperature at which metabolic rate is measured, which is important information when comparing the metabolic rate of ectothermic animals. BMR and body size. In theoretical treatise concerning homeothermic animals, Sarus and Remarques proposed what was called the surface law, which suggested that heat loss, the heat production of different sized species, should be related to surface area rather than the body mass. In mid-19th century, Regnault and Reset showed that resting oxygen consumption rates of endothermic mammals and the birds were 10 to 100 times those of ectothermic reptiles and amphibians. They also noted that mass-specific rate of oxygen consumption was smaller in larger species. Max Rubner, 1983, tested this proposition by measuring the heat production of dogs ranging in size from 3 to 31 kg and showed that the values were more similar when expressed relative to body surface area than if expressed per unit of body mass. This was regarded as confirmation of the surface law of metabolism. In 1932, two independent empirical studies showed that BMRs of different sized mammals and birds were proportional to the 0.73 power of the body mass. Broadley and Proctor, 1932, Kleber, 1932, rather than the 2/3 power of the body mass as suggested by the surface law. The expression of this relationship between BMR and body mass as a relatively simple power of function has seduced several scientists over the years into attempting to explain it by some underlying physical theory. Until now, none have successful, with the most recent being the supply side fractal theory proposed by West et al. 1997. This recent attempt has been strongly argued against the late Peter and colleagues, who pointed out that metabolic rate is not determined by supply constraints. A recent exposition of this fractal theory explanation that expands it to cells and mitochondria has ignored much empirical data on tissues and isolated cells that do not support this theory. The next slide is metabolic rate body size relationship. Is the one proposed by Hemmingsen, 1960. He suggested that observed exponents, and they are often very different in different groups of animals, result from an evolutionary struggle between two competing tendencies. Number one tendency is the tendency for metabolic rate to increase in direct proportion to body mass, as bigger individuals are made as copies of smaller individuals. And the second tendency is the tendency for metabolic rate to be limited surface constraints, mass to power of 0.67 during changes in animal body size over evolutionary time. As pointed out by Hemmingsen, if a rhinoceros had the same mass-specific BMR as a mouse, it would need a surface temperature close to that of boiling water to rid itself of the heat generated from its BMR. Such surface constraints are not restricted to loss of internal heat through the body surface of endotherms, as was initially argued in the early theoretical development of the surface law by the Sarus and Remarques, 1838, but include the surface limitations associated with the uptake and excretion of nutrients, respiratory gases, and metabolic waste, and thus equally apply to ectotherms as well. Composition of BMR and the membrane pacemaker theory of metabolism. Rolfe and Brown concluded that about 10% of the oxygen consumed during the BMR is consumed by non-mitochondrial processes. About 20% of the oxygen consumed is actually consumed by the mitochondria to counteract the mitochondrial proton leak, and the remaining 70% is consumed for mitochondrial ATP production. At a whole animal level, ATP production can be divided into 20% to 25% for sodium potassium ATPase activity, 20 to 25% for protein synthesis, about 5% for calcium ATPase activity, about 5% for gluconeogenesis, about 2% for ureagenesis, and about 5% for actinomyosin ATPase activity, and in the end about 6% for all other ATP consuming processes. These estimates are averages over the entire animal, and the relative contribution of the different processes varies between tissues. For example, it is estimated that although sodium potassium ATPase activity constitutes only about 10% of the cellular energy turnover in the liver, it is responsible for about 60% in brain, kidney. The next point is, it can be seen from these estimates that membrane-associated activities constitute a significant portion of resting metabolic activity, and thus BMR is higher in the animals. Regulation of BMR and thyroid hormone secretion rate. Cause or effect? It means that regulation of BMR and thyroid hormone secretion relationship. Does thyroid hormone secretion is the cause to increase the BMR, or it is an effect to produce more thyroid secretions in response to increased BMR? As described previously, it has been known for more than a century that thyroid hormones stimulate oxygen consumption. The early development of the BMR concept was closely linked with its use as a measure of thyroid status of individual humans. An obvious question that arises from this observation is whether it is the rate of thyroid hormone secretion that determines BMR. A common method of assessment of thyroid hormone secretion rate is to measure the rate of disposal of a small amount of exogenous radioiodine labeled hormone. As the blood levels of thyroid hormones remain relatively constant during such measurements, it can be assumed that the rate of disposal is equal to the rate of appearance of hormone in the blood. In this case, or in the case of thyroxine T4, this is the rate of secretion from thyroid gland, whereas in case of T3, it represents both the rate of secretion from the gland combined with the rate of peripheral production via the deiodination of T4. Tomasi in 1991 has collated disposal rates of both T4 and T3 for a wide range of mammals and demonstrated that they are related to a power function of their body mass not dissimilar to that which described the BMR body mass relationship. The secretion rate of T4 in mammals is related to the 0.81 power of the body mass, whereas the secretion production rate of T3 in mammals is proportional to the 0.74 power of the body mass. The fact that these parameters vary with approximately the same power of body mass says that there is a relatively constant stoichiometry between the consumption of oxygen and the disposal of these thyroid hormones. But what is cause and what is effect? In the body size related variation in BMR of mammals caused by the difference in thyroid hormone secretion production rates, or is it vice versa? That is, Are the differences in thyroid hormone secretion production rates between different sized mammals caused by differences in their BMRs? Not completely understood. This is not yet completely understood. But in order to understand above assumptions, we have to look at relevant data giving answer to following questions. Is there any difference? The first observation is there any difference in blood plasma concentration of T4 and T3 in different body size animals? The answer is, when we view the data suggests something else. The data suggests that there is no difference in T4 and T3 plasma concentration in different sized body animals. This implies that differences in secretion / production of thyroid hormones in different sized mammals is an effect of rather than the cause of size related differences in their BMRs. And the second assumption to test from the data is there any difference in BMRs with different plasma concentrations of these hormones, if any? Data suggests there are differences in BMRs in endo and ectothermal animals manifolds, even with similar concentrations of T4 and T3. So, the story is not still understandable or not clearly understood. This gives an assumption or a suggestion that there might be some other factors than concentration of thyroid hormones and body size that regulate BMR. But there is an agreement on this viewpoint that thyroid hormones are the regulator. BMR, is it a useful concept? The answer is both yes and no. To measure BMR is not particularly helpful if one is trying to precisely estimate the total food requirements of a species undergoing a particular activity. However, it is sometimes a useful concept if we are trying to estimate the increment in energy costs related to that activity. It has been particularly useful in comparing the rates of energy turnover in different sized animals and thus examining the influence of body size on an animal's physiology. Perhaps the last words should be those as said by Brodley in 1945. Basal metabolism is a convenient starting point for measuring the various heat increments as heat increments of fever, feeding, lactating, gestating or gestating, working, keeping warm in cold weather, and so on. So this was all about basal metabolic rate. Thank you very much.